Dear student, today we are going to discuss about the embryology or embryo formation. Embryology, if we want to define, then the study of animal development from the fertilized egg to the formation of all major organ system is known as embryology. Or in another word, embryology is the science that treats of the origin and development of the individual organisms. So, in simply, we understand that embryology is the study which deals the formation of adult organism from its fertilized egg. It is a gradual bringing to completion both in structure and in function. Its chief characteristic cumulative change in a progressive direction. All complexity arises from a single egg. So we have two basic views about the origin of complexity in the egg. I gave reference about the two basic views. These are preformation and epigenesis. Preformation. It tells that the gametes contain all of the elements which are present in the adult. Most biologists of the 17th and 18th centuries believed in preformation. Now we have epigenesis. This theory explains that the egg contains the material from which the embryo is gradually built. This theory became more popular in the mid-18th century. So, if we define both preformation and epigenesis simply, so we can say preformation, embryonic structure are performed within the gamete, either egg or sperm. And epigenesis, the embryonic structure arises anew from the interaction of substances within the gametes. I hope now you are able to understand or to differentiate both of these views, preformation and epigenesis. Now we can explain both of these views with the help of experimental basis. William Rox, 1888, and Hans Drech performed experiments to determine which theory was correct. They allowed a fertilized egg to divide to the two-cell stage. Rox's experiment. He used amphibian embryos. We know very well that frogs, toads, and salamanders are very well-known specimens from amphibians. He killed one of the two cells with a hot needle. The remaining cell formed half embryo. Rox called this half embryo as hemi-embryo. So, this experiment favors preformation. Drech. He used echinoderm embryos and well-known species from echinoderms, sea stars, sea urchins, sea cucumbers. He completely separated the divided cells. An entire animal developed from a single cell. It supports the epigenesis. Drech found that each cell retained the potential to develop entire organism. Biologists now know that Drech was correct. They believe that the killed cell was attached to developing amphibian embryos in Rox's experiment. It altered the development of the untreated cell. So, it was an important turning point in the embryology. Thank you.